questions to the end?
To the end.
Okay, that's fine. Can you guys hear me okay? Microphone's working? Great.
Not really.
Thank you, Piero, and thank you for inviting me.
Can you hear? Okay.
You can hear me okay.
The microphone is not doing a good job, but anyway.
Is this working? Probably need to turn it on later. Is that okay? If you want, I could use another one, but let me know if I'm not speaking loud enough. Yeah, it's a real honor to talk. I looked at some of the other talks, which were quite diverse and interesting. I'm going to talk about stem cell therapy for stroke and what I've done over the last quarter of a century with this. So many of you, I'm sure, have had relatives, family, or friends who have had strokes. Stroke is the sudden disability of a body function caused by disruption of blood flow to the brain. There are two types. The most common is what's called thromboembolism. That's where there's blockage in an artery in the brain. It can occur in situ in the brain. It can occur from the carotid artery throwing a clot up. A very common cause is from the heart throwing a clot. It blocks the blood flow. There's no delivery of oxygen and glucose, and simplistically, the brain cells die. The other type, which is less common, is bursting of a blood vessel causing a hemorrhage. Every year in the U.S. alone, 800,000 new strokes, and many more if you include Europe and Japan. 87% of these are the lack of blood flow, the blood clot type, called ischemic. And the only treatments acutely are clot-busting drugs, drugs which can dissolve the clots given intravenously, or now, and this is in the recent decade, putting a catheter up, threading it up from the groin, and pulling the clot out. That's a very good therapy, but only about 1 to 4% of acute ischemic stroke patients benefit from this. And there's no way to regenerate lost function in the brain. Most patients survive, but they are left with severe disability. 90% of the recovery after a stroke occurs in the first 6 months. After 6 months, even though patients have been through physical therapy, there's almost no recovery. So we have 7 million chronic stroke victims with disability living in the US, another 12 million in Europe and Japan, and most of these patients, 70 to 85% of them, have weakness, paralysis or partial paralysis. More than 50% of these patients are functionally dependent, actually, in activities of daily living, and it costs $130 billion per year in the US alone. So very, very important if we can develop a therapy to improve motor function, and I'll talk about speech as well. You know the signs of stroke: weakness, paralysis, blurred vision, difficulty speaking, problems with balance, falling. It can occur with a headache, personality changes, difficulty swallowing. With the exception of vagal nerve stimulation and intensive physical therapy of the upper limb, that's putting a stimulator on a nerve in the neck, simple operation. You must do very intensive therapy of the upper limb. That was approved in 2021 by the FDA. It has a very modest improvement in weakness of the arm. So this is still really an unmet need for chronic stroke patients. What about stem cell therapy, which is becoming popular? We started this 25 years ago, and in the lab we transplanted human neural stem cells into rat and mice brains that have been stroked, and looked at the effect of the cells on the brain, but also the effect of the brain on the cells, because there's two-way crosstalk. And we found when we placed the cells, here's the stroke, too close to the stroke, they didn't survive. It's a very inhospitable environment because there's no blood flow, a lot of the tissue is dead, but when we move the transplanted cells just a few millimeters away, they not only survive in large numbers, but they migrate to the stroke. They're stained brown here. So there's targeted migration, they can move. Why do they move? It's interesting because if you put them into a rodent brain that does not have a stroke or any other injury, they stay put. They don't move. They move because, and we showed this and others have shown it, the stroke environment secretes chemicals, they're called chemokines, they're molecules and proteins that attract the cells because these stem cells we transplant have receptors on them that interact with these chemicals in the brain. And we're counting on the fact that these stem cells are smart. So the important thing is not just they migrate to the edge of the stroke, but that they recover function. And you can see here in various types of tests, the cells in red compared to the buffer, which is no cells, show increased recovery if you transplant them after the stroke in rats and mice. So, how do they work? The initial notion, of course, was that these cells we put into the brain turn into the different types of cells in the brain, neurons, astrocytes, and oligodendrocytes. And we thought that when they turn into neurons, they reconstitute the circuits. That is not how they work. The way they work, we were one of the first to discover, is that they secrete very powerful trophic factors, growth factors, molecules, and proteins that enhance native recovery through all these mechanisms, which are endogenous or native mechanisms. And in a sense, what they do is turn the adult brain into a neonatal or an infant brain, which has a lot of what we call plasticity. So if a 1-year-old has a stroke and they're paralyzed, they usually make a complete recovery. And when my granddaughter, who's a few years old, cuts herself, by the next day it's healed. When I cut myself, it can take a month to heal completely. So we think simplistically we are increasing the plasticity of the brain and turning it into a younger brain. And perhaps the most important is modulating the immune system. So we've done lots of studies in the lab. I won't go all over those, but about a decade ago, we started doing a study with cells from a company called SanBio. It was a local company in Mountain View. They're actually based in Japan. And what we did was take cells. The company took bone marrow cells from donors. 2 patients provided enough cells for 18 patients. The cells were taken from the bone marrow, put in culture and expanded and cryopreserved, and then shipped to the sites. And it was 2 sites, Stanford, where I did the transplant, and University of Pittsburgh. And what we did was to put on what's called a stereotactic frame. It's attached to the scalp and skull, and it creates a 3-dimensional framework, like a GPS system. So you all know GPS because you navigate in your car with GPS, which has an accuracy of 20 feet or so. When we use this on the brain, it has an accuracy of less than 1 millimeter. And so we can put the cells exactly where we want, and we target the deep stroke, which is schematically located here, and we put the cells around the stroke, not in it, because I told you the cells don't survive even temporarily there. And these patients were 33 to 75 years old. They were at least 7 months to 3 years out from a stroke. You knew they weren't going to recover, because they've been through physical therapy, and patients don't recover after that period of time. And it was a safety study primarily, but we did have a primary efficacy endpoint, which is a certain scale. And this is what the strokes look like. So you guys are familiar with MR scans. This is a cross-section of the brain like this, looking up from the bottom. And the stroke, these white spaces in the middle are the water spaces in the brain, so you can ignore them. But the white areas outside of that are the stroke. And these patients had to have a deep stroke like this, but they could also have a cortical, more superficial stroke, but we targeted this deep stroke, even if they had a cortical stroke. The patients were treated without putting a tube down and intubating them, without general anesthesia. They were sedated, and we numbed up the scalp and gave them nice medicine so they didn't remember much of the procedure, although they were awake. And we did a dose escalation. I treated 12 at Stanford, 6 were treated at University of Pittsburgh, and I put in a small hole in the skull, opened the membrane covering the brain, and put a needle through and injected the cells around the stroke. Patients all went home the next day. And we found out it was safe, no serious adverse effects that were related to the cells. Patients did great, and we were happy about that, but we were shocked to see the patients recover compared to their baseline. So if you look at the average of all these patients compared to their baseline, which would be 0, the patients started improving with their motor function, arm or leg, at 1 month up to 3 months. It was stable at 6 months, sustained at 12 months and 24 months. This was the primary outcome, highly significant, and on other scales of motor function, also highly significant. Big shock. And in fact, on this scale, if you look at what is considered a meaningful recovery, that would be 10 points or more on one of the scales, the motor scale, out of 100. Physiatrists and physical therapists and neurologists consider this. It means that patients can do things they couldn't do before. They can turn a doorknob, they can eat, they can walk independently. 3/4 of the patients achieved that, and it occurred early on. Let me show you an example. Here's a patient, 71, and I thought 71-year-olds couldn't recover. They're way too old. Although I'm 73, now I think 71 is, if I could be 71 again, I'd be happy. This patient is 2 years out after a stroke after a heart valve replacement. And this is before the transplant, and this is a neurologist examining her, and she has no motor function. She can do nothing on her left side except move her thumb, wheelchair bound. So here's what she can do.
Can you extend it at all?
All she can do is move her thumb.
Move your thumb.
Can't move the arm. This is what she can do with her leg.
1, 2, 3, 4, 5. Let go.
That's it. Hasn't been out of a wheelchair for 2 years. So we transplanted her, and I want to show you what she looks like at 3 months after the transplant.
As hard as you can, the arm.
To your nose. Oh my goodness.
Video, you do that?
Touch my hand. 1, 2, 3, 4, 5.
So her son is hugging me, and this is a miracle. So I called our neurologist, who you saw examining her, and neurologists are appropriately skeptical. They're not surgeons, and they're very smart. And unlike surgeons like me, who tend to be enthusiastic and over-optimistic, I said, "You're not going to believe this," but she's lifting her arm up, kicking her leg off the bed. He said, "I don't believe you." So he said, "Come over and examine her." He examined her, and he said I can show this email. So when a neurologist sends you an email with the subject "I believe" and says, "I must admit it's a bit of a miracle," you know it's likely not a placebo effect. So here she is 6 months later. She's walking after being wheelchair bound for 2 years.
Great. Okay, smile.
Here's another patient, younger. This patient had a stroke 2 years prior. We knew she wasn't going to get better. In her left brain, that controls the right side of the body, but it also controls speech. And she was unable to move her arm. Her speech was so bad that you couldn't understand her. She couldn't order food over the phone. And her walking was so difficult that she did not want to get married. She thought she'd be embarrassed trying to walk down the aisle. And here's what she looked like before we transplanted her 2 years after her stroke. Can't get the arm off the bed. Okay, here she is 2.5 months later. And she actually gave me an award 4.5 years after her transplant at the Smithsonian.
For 2 years after that, I was unable to say more than 20 words. I couldn't move my right arm more than a few inches. I could only walk for 5 minutes without needing a'It's been 4.5 years to the day now. and I'm able to climb stairs, have conversations with family and friends. I run, I work out. My life is amazing. I was also able to become a mother.
So she got a lot of press because some of these videos went on YouTube, and you know what social media it does these days. And someone sent me this video. This is same woman, 10 years after her transplant. She's in Italy, and she's climbing a wall. This is someone who couldn't move her arm, couldn't walk well. So what we learned, we thought that those circuits after a stroke, 6 months or years later, were dead. They're clearly not dead. So this changed our notion of what happens after a stroke, and probably after other injuries like spinal cord injury, traumatic brain injury, maybe even after degenerative diseases like Parkinson's, Lou Gehrig's disease, maybe even Alzheimer's. And the question is, how do we resurrect those circuits? We're still trying to figure that out. So in my lab 25 years ago, we developed a neural stem cell that was shown to be beneficial in 2 independent labs, 2 stroke models, recovered function, doesn't cause tumors. And it took me 25 years and more than $50 million in grants and philanthropy to get it through the FDA and complete this trial, which we just finished 1-year follow-up on. This is the first in human trial done at Stanford with cells developed at Stanford. That's unusual. Usually we're great at discovery at Stanford. We haven't been very good at translating it until recent years. And again, patients were in this age group. They were years out in general from their stroke. You knew they weren't going to recover. We did the same thing that I showed you before, transplanting the cells on a dose escalation. By the way, there's no dose response. All the doses worked. There was no difference. And we did immunosuppress these patients for 8 weeks. We don't think we need to do that. And again, we now use this Fugl-Meyer motor score of 100 points compared with their baseline as the outcome. And to my surprise and everyone's surprise, we saw spectacular recovery in many of the patients. So on a 100-point scale, 100 would be normal. Patients vary. This patient was pretty good, and I thought he's too good, he's not going to recover, but he recovered 19 points. Remember, 10 points or more is meaningful. And all of the patients improved some. These patients down here, very severely affected, 3 of the 4 did not improve much, and all of these patients would be excluded from the next trial we're going to do, but I'm going to show you one patient who surprised us. 2/3 of the patients at 1 year had a clinically meaningful recovery, and here if you average all the patients compared to their baseline, so it would be 0 starting, again they started improving at 1 month, they increased 2 months, 3 months, they even increased more from 6 months to 12 months, which we didn't see in other trials, and statistically highly significant, and it reaches 11 point, almost 12 points, more than that 10-point cutoff, that threshold I told you. If you look at the gait speed, this is how long it takes to walk 10 meters, so lower is better, and we saw an improvement of almost 10 seconds in the gait speed overall. 5 patients could throw away their canes, 2 improved from a hemi-walker to a cane, 1 from a hemi-walker and belt to no belt. And Barthel Index, which is a measure of activities of daily living, it's an important measure, improved again significantly, quite a bit within 12 months. I'm going to show you some patients again. This guy was 7 months out from his stroke, been through rehab, and before surgery,
Put up as best you can.
after his stroke, he cannot move his arm at all, no movement in his hand.
All right, this one, can you get it up off the bed?
He has no hip flexion. He can barely get the leg off the bed.
Just get off the bed.
And before surgery, even though he can't lift his leg, he can walk with a cane because he has what's called spasticity. It's stiffness from the stroke. So he can swing his leg, even though he can't lift it, but very slow. Here he is 3 months later. And look at his leg. Now he's holding it up for 10 seconds. Look at him at a year, no cane, very fluid. Look at him at 18 months. He's now doing pull-ups, and he's riding a bike. 2 years, this is something I never thought we'd see because the hand function is very difficult to recover. He had no hand function, and he's eating yogurt out of a cup with his disabled hand. His wife is impressed. He's swimming. In fact, at the 3-year follow-up, he couldn't come back in person because he moved to Atlanta and started his own company, so couldn't take the time off. This patient I want to show you because we didn't measure speech, but we're going to do that in the next trial. This is following her stroke, but before the transplant.
Just read these sentences.
She's also partly paralyzed.
Sit down. 50-50. Next. Very, very good.
Okay, very speak, but very slow. Look at her 2 months later.
You know how down to work. I got home from work. Here, that big one.
So we think language is something we can improve with this kind of transplant. And this guy's interesting. This was one of those lower level patients. No movement at all in his arm or hand, nothing. And here he is, years out from a stroke. Before the transplant, he cannot move his arm at all. Nothing. And at 1 month, nothing. At 2 months, nothing. I thought we'd never see recovery. And at 3 months, he did this. And although it's not a lot and functional, neurologists and physical therapists think this is another miracle, from nothing to this. I saw him today for his 2-year follow-up, and he is still improving. And in fact, if you look at 2 years, 70% of the patients who are out 2 years still show clinically meaningful recovery. And you can see patients are improving between 1 year and 2 years, which is remarkable. We never saw that in any of the other trials. So, my belief is that this brain transplantation with the cells in chronic stroke is safe and well tolerated. There's no real treatment for this. We're seeing recovery at 1 month increasing to 24 months. We're starting another trial. We're trying to move quickly. This is going to be multi-center. We've already got 13 sites in the country. It's going to be blinded so that some patients will get the cells through the burr hole. Some patients will only get a burr hole, a little hole. And no one will know except a few people in the OR. The patients won't know, and the people, the physicians and therapists evaluating the patients won't know what they got. And we hope to do this quickly. We're going to use one arm. We're going to add an exploratory arm where they don't get the immunosuppression. We don't think we need it because, as I mentioned, the cells are immunosuppressive. They modulate the immune system. We want to get through this phase 2B study quickly and move to a phase 3 because I would like to see this approved by the FDA for all patients in my lifetime. So we still don't know what the best cell source is, the type, the number. When should we treat? Originally, I thought it would be best to treat subacutely within 1 week to 3 months. That was where we saw recovery in animals. But now I'm convinced chronic is better. That's where we have many more patients. And the problem with treating too soon is that patients are recovering, as I say, for 6 months. So you don't know whether it's the cells or the natural recovery. Should we deliver them into the brain like I am, or through intravenous, through an artery in the brain, through the canal in the spinal, through a lumbar puncture, spinal tap? And now, people in the 1990s when we started this, people laughed at me and they criticized me. They said, the science isn't there, you should not be treating patients. We were one of the first groups, but I said, if we can do it safely, we learn so much from the patients you can never learn from animals, which proved to be true. We learned about mechanisms from patients that we didn't even have an idea of studying this aspect in animals. And now, of course, there are 72 trials ongoing or unpublished. There's another 40 which have been published. Most are using bone marrow-derived cells, only 15% the neural stem cells, hematopoietic or another type of blood cell. And most are doing it intravenous because that's easier, some arterial, and only 20% into the brain. I want to thank my team at Stanford who have been with me, some for 30 years, my colleagues at Stanford and elsewhere, and the clinical team who did all the work for this. And thank you for your attention.
So let's start with a silly question. What is missing? When will we have this procedure like standard? You have a stroke, they take you to the hospital, you get the Steinberg procedure, you go home within 1 month, there is progress and so on.
Yeah, so remember, this is for chronic stroke patients. We're not going to be doing this when a patient comes in with an acute stroke. And one of the reasons we won't is after acute stroke, there's a lot of inflammation in the brain. It's not a good time to be putting in a needle and injecting anything. And some patients recover over 6 months. So we're targeting this for 6 months to now the next trial is going to be up to 7 years. But maybe it's appropriate to give intravenous stem cells. And that's what several companies have targeted. Unfortunately, although there was a lot of promise and enthusiasm, none of the intravenous trials and none of the intra-arterial trials have panned out in the phase 3 studies. Now, we're not in phase 3 yet, and a lot of therapies fail between phase 1 or 2 or 3 as we move along. So we're excited, but we don't want to get too excited yet because we have another 2 studies to do. But if we can achieve these kind of results in the next 2 studies compared to control, then we will have a new therapy for an unmet need.
And in one of the last slides that you showed this diagram, and you showed that the most used cells are actually the MSC, not the neural cells. Can you say something about this MSC, which I can't even pronounce what they stand for?
Mesenchymal, yeah, mesenchymal or bone marrow derived, or there are blood cells you can use. It may not matter which kind of cell you use. As I say, we don't know what the best cell type is yet. The way they work is they don't integrate into the brain, as I said, and turn into neurons. So they secrete these very powerful molecules and proteins and other substances that enhance native recovery. We think the neural stem cells that we're using may be better because that's what the brain is used to, but it's the secreted factors that are important. And the cells we have found could secrete dozens, if not hundreds of factors. And the advantage of putting the cells in instead of just giving the factors is severalfold. One is you can't get the factors into the brain because they don't, if you inject them intravenously or into an artery, because they don't pass through the barriers. Secondly, as I alluded to, the cells interact with the environment. It's a two-way crosstalk. So the stroke brain, even chronically, says to the cells to secrete this amount of this protein, this amount of this molecule, and that may be a benefit, the crosstalk. So we still have lots of unanswered questions. It's still very new. It's a new therapy. But I predict over the next decade, we're going to see stem cell therapy generalized for various types of neurologic diseases.
But you feel that neural cells is the right way to go? Or you think
we don't know. It's a good question.
Lots of questions. Shout or take mine?
I think I can speak loud enough. So, can you not get enough statistics without doing the double blind? It seems like you're so close to an ethical problem there of putting people through that. You understand exactly what I'm saying.
I do, because one of our investigators, so I contacted 36 of my colleagues, and I've been presenting this stuff, about whether they'd be interested in participating in the next study. 34 of the 36 immediately said yes. We can only have 10 to 13 sites, so we disappointed a lot of people. But one site we contacted, the primary investigator said, I think it's unethical to be doing this. But it's not, because you have to do studies. There's a placebo effect, and we don't know for sure it's the cells. We think it's we think it's very unlikely.
Do you think there's a placebo effect going on with this?
There can be. I mean, surgery is a very powerful placebo. I don't know. Some people think it's just the needle. We don't think it's just the needle, because in the lab, if we use the needle with no cells, just the buffer, we don't see the recovery. But you have to do controlled studies. I mean, that, unless, as many of the new administrative officials feel science isn't important, many of us still think that science is important, and you've got to show in controlled studies that it is important. In fact, with our next trial, if we did a statistical analysis, and if the next trial is as powerful in effect as this one, even assuming that the control patients who just get the hole and no cells recovered 25% of the treated patients, we only need 11 patients in 2 groups to show the effect. That's how powerful it is. But you know, I think we would have a hard time. Some people say you should have a control where you just put the needle in and inject a buffer, but that we'd have a hard time getting through the institutional review board. I agree with you, but as a scientist, and the FDA would never approve this therapy without another.
Right, so that's the real reason.
Yes, for the FDA.
There was a question back there.
Did you measure cognitive change, and how did that get impacted for these people?
Yeah, we did not measure it. It's a great question. But one of the earlier studies with some other cells did see some improvement in one of the cognitive tests. So we have some tests which grossly measure it, but when you start this, you want to have some kind of outcome that is very measurable. Cognitive function is much harder to measure. So that's why motor function is easier, and we're going to also measure language function with a language test in the next trial, because you saw some of the benefit on language.
Thank you.
Great question.
I think he was first.
How hard is it to extract the stem cell from the bone marrow?
How it's difficult? Simple. You just put a needle into the hip. It's a little painful, but very easy.
So in terms of are you looking for specific markers on the stem cell?
Yeah, that's a great question. So those cells were processed in culture, and they actually were transfected with a gene that helped with the propagation in culture, and that's one problem. Our cells are not manipulated genetically. All the other cells that have been transplanted into the brain have been genetically modified for various reasons. So we don't know the answers to all that, and which cell is the best type. It's easy, and remember, those aren't from the same patient. They're called, they're allogeneic, not autologous. Our cells are, we made a master cell bank. So our cells actually were embryonic derived, but we differentiated them so they're not embryonic stem cells. We differentiate them so they don't form tumors. Then we made a master cell bank, and now we've made a working cell bank at different passages because we don't want to deplete the master cell bank, but we have an unlimited supply basically. So those bone marrow cells, they have to be derived for each trial. They don't have a master cell bank. That's another advantage of ours. That's a great question too.
Yeah, so it seems like you're seeing gradual improvement over a long period of time. Have you run it long enough where you've seen that flatten out, and would you consider doing a second treatment?
Yeah, I get that question a lot. In the SanBio study with the bone marrow derived cells, it increased to 3 months and then plateaued at 6 months, 12 months, 24 months. With our cells from my lab, we're seeing improvement, as I showed you, even from 1 year to 2 years. We have anecdotal evidence that there's even improvement with 3 years, but we think that physical therapy and increased activity is important, and we encourage that. We didn't require it in this study, but we think that the activity may work synergistically with the cells. in reconstituting the circuits or invoking circuits that weren't working before.
What about doing subsequent treatments?
Yeah, so we haven't done that, but everyone, even some of the patients who have improved, said, I want another treatment. But we haven't done that in the lab. We probably need to. I mean, there's a lot of work to be done. Whether a second treatment would be useful.
Bigger question. While as you are studying how to heal stroke, are you learning something that could be applied to cognitive decline that happens?
Yeah, we think there are similar mechanisms. I mean, Alzheimer's is very much a vascular dementia. We think, in fact, there are some studies being done in traumatic brain injury. That Sam Bio study that was positive, they did a subsequent study in stroke and also in traumatic brain injury. The traumatic brain injury was more positive, so they decided to go with that, and they got approval in Japan using those cells to treat chronic traumatic brain injury. They're trying to get approval in the US, it's much tougher. So yes, I think it's going to be because of similar mechanisms. It's all about circuits and how to resurrect circuits that we thought were dead. And circuits are the problem in injuries, stroke, traumatic brain injury, spinal cord injury. Circuits are the problem in degenerative diseases, Parkinson's disease, Lou Gehrig's disease, ALS, probably Alzheimer's. The circuits aren't working well. So yes, we think once we get it approved, I'm sure once we get it approved for stroke, it'll be generalized because you can use it off-label in other diseases.
One more?
Question, yeah. There's a lot of research being done on stem cell therapies for inherited retinal diseases, and I'm wondering whether you are involved in any of that or not.
No, but some of the companies, some of the companies that started with stroke ran out of money or ran out of resources during COVID and couldn't complete the trials that were looking promising, and they focused on macular degeneration as an example. So a lot of interest there because it's easy to get them in. Because you can inject them into the vitreous directly. You don't have to go,
Right.
into the retina, and you don't have to go intravenous or
It's also a
intraarterial.
privilege.
Yeah, no. So that a lot of companies are working on that. Yes.
Excellent.